Now, as the smart board here fires up, this should be the last one we talked about yesterday, dealing with RNA primase. RNA primase right here, okay, the pink, and the small RNA primer, okay? This RNA primer is what starts the whole process, okay? After the DNA helicase, and I said for this particular picture, this green one here is DNA helicase, all right? It's DNA helicase there. It is unzipping the stranded DNA, okay? We need a primer to come along to start adding on, okay? And you're going to see here the 5' to the 3' is going to mean something here in a little bit. What did I tell you to call this thing right here? You have an arrow right here, right? What did I tell you to call that? The replication fork, okay? The replication fork. So on our third diagram now coming up here, the DNA polymerase adds the appropriate nucleotides to a chain to the 3' end of the parent strand and works towards the 5' end, the replication fork. Okay? So, if we go back and look, what that means is we're going to have DNA polymerase. See this where it says 3'? It's going to add it to the 3' end, so it's going to end up working this way towards that replication fork right here. Okay, that's what this is saying here. Add to the 3' end. So, this makes a new DNA strand in the 5' to 3' direction that we call the leading strand. We call the leading strand. Now, how can you tell whether something is the leading strand, or you're going to learn here in a bit the lagging strand? The leading strand is moving towards the replication fork. Okay, so flip open that little box there on your flip box, so the third one. Flip open your flip box. See what you have there? Here is the diagram. Okay, here is the diagram that I want you to put on there. So I'll give you a little time here to add that on. Okay, I don't want you to do this part yet because that is the next one. Just do that top part. So now, folks, what this is signifying, let me get this off. I didn't want you to write that down yet. Okay, I wish I wouldn't have all of this in here. We are only going to be discussing right now this part right here. Because we said that this DNA polymerase is adding, and the big thing here is the leading strand. The leading strand of the DNA as it replicates is one continuous strand. Okay? One continuous strand, and you can tell by this arrow right here, what way is it heading? It is moving towards the replication fork. Okay? It is moving towards the replication fork. So, that is the leading strand. Now, this next one we're going to talk about is going to deal with the other one, the lagging strand. The lagging strand. So we have the leading strand, we have the lagging strand. Now, nucleotides are added in chunks, and those chunks we call Okazaki fragments. Okazaki fragments. The Okazaki fragments tell us which one is the lagging strand. Now, if you look right here, okay, I didn't mean to switch it, okay, the Okazaki fragments are on the lagging strand. Okay, now, if you look here, the Okazaki fragments, and what you should draw under that flip box, which is flip box number 4, right? What you should be drawing under that is this stuff right here. Okay, that stuff right here, because that is showing you the Okazaki fragments. All right, now, if you notice, I'll give you a second to write this down before we talk about it. If you notice here, these are discontinuous. They are fragments. And we have little pieces right here on the ends of these, folks, that are also primers. We have the RNA primers on each one of the Okazaki fragments, okay? Because that starts the adding. Once again, which way is it adding to? It's adding onto the 3' end, just like with the other one, but this time we are working away from the replication fork, correct? So, when we're going away from the replication fork, we have the lagging strand. Which strand is going towards the replication fork? The leading strand. The one that's going away is the lagging strand. And you can always tell the lagging strand because it's put together in what? Okazaki fragments. Okay? That is named after the scientists who discovered these fragments. Okay? Okazaki fragments. So, when you flip up the flip box on number 3, you should have the leading strand, correct? When you flip up box number 4, you should have the lagging strand with the Okazaki fragments. Now, we've got these Okazaki fragments, we got these leading strand, we got the lagging strand, right? Now, let's go into when the entire new strand is replicated, DNA polymerase removes the RNA primer and fills it with DNA nucleotides. So, as you can see here, here is the RNA primer, that little red section that we talked about, okay? I'll let you go ahead and draw this. Why don't you color this one in too, so you can see the colors. Yours is black and white. Why don't you draw this in and color it? Okay, let's talk a little bit about this, okay? We've talked about DNA polymerase. Now, just something, you do not need to be able to distinguish between the two, but I'm just showing you here that we have DNA polymerase 3, DNA polymerase 1. They are different. They are different. This DNA polymerase number 3, DNA polymerase number 3, is the one that is adding the new nucleotides to the chains, whether it be leading or lagging. All right? DNA polymerase 1 is a different type of polymerase. Its job is it comes in and it removes the primer. Okay? Right here is the RNA primer, that red part. Can you see that polymerase right here is removing that red part and putting DNA strands onto it? Okay? So, in this step, the DNA polymerase takes that RNA primer out of there. Now, you're going to learn a little bit later. We haven't talked about RNA yet, but it is different than DNA, isn't it? Okay? Anybody know anything about RNA? Okay? We'll talk about that later on. There are 3 types of RNA. RNA is different than DNA. RNA's only got a single strand. DNA has a double strand. Okay? RNA has uracil, which replaces thymine. So you won't find any thymine on RNA. Or excuse me, yeah, on RNA. Thymine's only on DNA. Uracil replaces thymine in RNA. So they are different. So this strand, this RNA primer, is filled in now with DNA. Okay? So this polymerase comes through and fills the RNA in with the DNA nucleotides. Okay? So you can see right here, okay? You can see right here, like I said, that little red primer is being filled in with the light blue nucleotides of DNA. Now, our final step is right here. DNA ligase links the 2 sections. Okay, DNA ligase links the 2 sections. And go ahead and color this in. Go ahead and color this in, and we'll talk about this one too, because there's a lot of stuff going on in this one. All the good stuff going on here. Okay? All the good stuff going on here. Number 1, okay, number 1, we can look and it's labeled the lagging strand, right? So when we think of lagging strand, we think of what? Okay, what did I tell you you could tell the lagging strand by? It's just a couple of slides ago. Okay, the way that it's going one way, it's going away from the replication fork. What else will you see on the lagging strand? Made up of chunks that we call Okazaki fragments. Okay, so this is the lagging strand here. I can see, here's one, because look at that little gap. Here's one, there's one, there's one. Here's the start of another Okazaki fragment. So here's a fragment, here's a fragment, here's a fragment, there's a fragment. We got Okazaki fragments coming in there, right? So I'm seeing those Okazaki fragments. So I know that this is a lagging strand. Now, DNA polymerase 3 right here is adding on to the RNA primer. I can see my RNA primer right there. Now look at this one. Here, the RNA primer is being switched out, isn't it? It's being switched out for DNA by the other DNA polymerase number 1. Okay? Those are all the steps we just talked about. Now the final step, once this DNA polymerase number 1 gets all of that primer changed out, look at that right there. Do you see that little gap? Do you see the little gap in between the 2 Okazaki fragments? Okay? DNA ligase will come in and it will bridge that gap. So DNA ligase comes in and this gap will no longer be there. The ligase will bridge that so that now we have one continuous chain of DNA. So there's a bunch of little things that are going on in this. Okay? Bunch of little things that are going on in this. Now, what I would like for you to do, okay, on this particular slide right here, we talked about this yesterday. I forgot to get this in your notes, so I made this yesterday after we got done discussing it. Now, you should be able to fill in the notes here based on what I have up there. Your top one says the blank strand is preserved and the blank strand is assembled. The parent strand is preserved and the daughter strand is assembled. The second line talks about semi-conservative replication, which results in DNA strands that were identical. All right? That were identical. Now, you have a drawing up on top of that. What I want you to do, okay, your drawing looks something like this. What we're going to do, folks, is you're going to make different colors than what I have up here. So what I want you to do is I want you to color this top one red. So color this top one red like I have up here. They're both the same color. So color that one in red. What I want you to do then is, and I'm going to move my little box here, up here. What I want you to do then is I want you to color every one I have up here that is dark blue in red. Because the red ones are the parent strands. So color all of these blue ones in red. Then, just so we have a different color to contrast that, I want you to color all the light blue ones in green like I have here. Now, as we look at those and explain semi-conservative replication, notice, okay, that we have two red ones up at the top. That two red ones split, okay? So those are the parent ones. We split, and now we have a daughter that pairs with each one of the parent ones, okay? So the green one is the daughter, the red one is the parent. Right here, they have split, right? So we have one red and one green. But what can you tell me about those two strands of DNA? They are what? Okay, those two strands are identical, right? Because we've replicated the parent strand. Now, down here, these two have split again. This time, the parent strand is still here in red, okay? But we've also duplicated the daughter strand, right? But nonetheless, even though we've duplicated the daughter strand, they are still what? They are still identical because we are just copying the parent strands. So that is semi-conservative replication. All four of these strands are identical, and they all were made off of the red strands that we still see there down at the bottom, right? We understand that? That is what we mean by semi-conservative replication. Okay? Now,